Good afternoon or good evening, depending which part of the world are you um, logging in. So we are going to start the activity. Unfortunately, our keynote speaker is having some difficulty in getting connected. He connected once, but then the line dropped. I think he's looking into it. So in the meanwhile, so he joined. Wonderful. So I welcome all of you. And let me introduce one by one the panelists over here. Dr. Umar Chughdai, I think most of you may be knowing him from his background in US because he is a trained pathologist from US. And then he moved back to Pakistan to serve the community as such. A King Edward Medical University graduate and having a fellowship in surgical pathology. And he has the command of running the largest private sector lab network in the independent lab work with the 325 branches all across Pakistan and 10 dedicated testing locations to be more reactive, supportive, and facilitative. So he is the person who is very close to the line of action from both angles, the diagnostic, and recently they ran the immunization services as well. So after Dr. Hamid Merchant talk, we will request Dr. Umar Chughtai to share his experience and how they are planning to take it to the next level and how to use that data for the development of science. And our other panelist is Professor Akhtar Hamid, who is a pediatrician and assistant professor with University of Toronto, and also the president, Canada Pakistan Research and Development Council. So we welcome both the panelists, and Dr. Akhtar Hamid hopefully will share his experience about the most vulnerable and the loved one community, the pediatric population. So what is in the pipeline and what are the possible dates really to have more insight about the pediatric immunization? And the author, the chief architect, and the person running the whole planning the show, Professor Dr. Babar Rao. So we welcome. So I'm so excited because he was so busy and I don't know how he spared the time, really managed to join the session once again. So it's a great thing. Now, I would like to introduce Dr. Hamid Merchant. He is a subject lead in pharmacy with the University of Huddersfield, UK, having over 18 years of experience in pharmaceutical research and development, which include both industry and academia, and involved in the research design and creating evidence synthesis for the safe use of the pharmaceutical and other intervention to address the pandemic. And he spent quite some time with the University College London, but basically a pharmacy graduate from Pakistan. So we welcome our panelist, our chief architect, and our keynote speaker to today's session. Before formally starting the session, I thought just to set the stage, really to let you know what was the motivation to have this emergency webinar, because usually we have the routine structured seminar. As we all know the impact as of today, you know that globally the number of cases has crossed 147 million, and unfortunately so far there are over 3 million reported deaths around the globe. And here in US, 226 million doses has been inoculated to the population at a rate of 2.82 million per day. Back to the homeland, Pakistan, the situation, the stage over there. So just yesterday, the reported cases, they were staggering between 5,600 to 6,000 based on the number of tests, which are little less than 60,000 and showing a positivity rate of over 11%. And unfortunately, one district in KPK reported yesterday a positivity rate of 80%, 80%. So the number of confirmed cases so far has close to 800,000. And so far, the reported deaths in Pakistan, they have crossed the figure of 17,000. And the number of critical cases, they have crossed 5,000, which translate it to the pressure on the services in the hospital and the intervention which is mounting. So that was the disease scenario back in Pakistan. And about the vaccination scenario, just a snapshot. So far, 5 vaccines have been given emergency use authorization in Pakistan. 2 are inactivated viral vaccine, Sinovac and Sinopharm from China, and 3 vector-derived vaccine. Vector-derived vaccine, we have the Sputnik, which is the Gamaleya Institute from Russia. Then we have the CanSinoBio, a single-dose vaccine from China. They conducted their clinical phase 3 trial in Pakistan on 18,000 people. And the last one is the Astrazeneca, which has been authorized, but so far no supply has been received from the COVAX. So factually, there are just 4 vaccines which have been injected to the people community in Pakistan. And we have inoculated back in Pakistan less than 2 million doses, which mean it is going to cover less than the 1% population. And on a daily basis, we are injecting 9,300 doses. That's the figure for today. And, you know, considering the eligible population over the age of 18 years, their number is staggering over 100 million. And with the present arrangement, there are some estimate that it may stretch up to 2023 to cover 70 or 60% of the population. And from the supply side, uh, over the last 24 hours, we have received about 1.5 million doses. And we have a firm commitment for 35 million doses, which we see to be supplied in May. So that is a very quick overview of the disease prevalence, about the availability of the vaccination. And the last small addition, so far in the private sector, uh, 50,000 Sputnik vaccine has been allocated and it is available. The second shot is in progress. And 10,000 CanSino bio vaccine is going to be available from this week to some designated institutions. So that is a very, very quick overview to set the stage. And we are here really to understand exactly about the vaccine hesitancy, the safety of the vaccine, which is a critical factor. Here in US, just yesterday, it was resumed from the J&J, Johnson & Johnson. So we would like to know exactly the scientific evidence behind that. Sometimes there is a changing point, the age limit, particularly with the AstraZeneca, there were so many concern and there is a changing here in Canada, there is a changing dynamics in US, there is a hesitancy from the scientific community and from the advisory bodies. So it took quite long to uh, give you an insight about this thing. So all this, really, we welcome, and I will request Dr. Hamid Merchant to please start. Please go ahead, Dr. Hamid Merchant.
Thank you very much, Dr. Uzair, and thank you very much for all the panelists here, the APPNA Foundation, CPRT, and for all of you to come over to join the talk. I'm going to load up my presentation, and give me a sec. There you go. Can you just confirm that if you could see my screen?
Yeah, you are perfect.
Brilliant. So today we're going to talk about particularly the recent thrombotic events, which has caused lots of anxiety and confusion among the public and healthcare professionals as well, and contributing to vaccine hesitancy, as Dr. Zair was explaining. So you're going to look at what are the events, how the pharmacovigilance system is working to pick up the early signals in the pharmacovigilance, what we have missed, what we can look at the moment. Are there any mechanisms which can explain why and how the clots have been forming in those minority of subjects? And are there any ways that we could have identified the high-risk population who might be at a higher risk of developing clots? If we could shield them, we might be able to prevent those minority of severe adverse event in the population, which could improve the hesitancy in the other way. If nobody would die with a vaccine-related side effect, that would be a good outcome we would like to achieve in the minority. So to do that is a very difficult process. There's lots of unknowns and unknowns, which we have to learn, and lots of scientists, particularly those who are in coagulation research, are putting their brains into some fundamental understanding of the coagulation pathways mediated by the vaccine. So I'm trying to summarize a few mechanisms which we have seen so far, have been proposed, some of the work which we have done personally, and some of the work being done in Germany. And the talk is intended for the healthcare professionals. So I'm going to talk a few things quite quickly. If I'm passing through quite fast and there were some questions, please hold on to them. At the end, I'm happy to take them and answer them. So before I start, just a bit of disclaimer that I have not been funded by any organization which would affect the ideas and the concepts being presented today. It is the clinician's judgment which is important to make to give an advice to the patient. Anything given in the presentation is not a direct advice, and it has to be personalized based on patient's history and what you see in clinic. To give a perspective, I'm not going to go in a lot more details about COVID vaccines, but just to give a bit of background to make sure you understand the mechanisms when I'm relating it back. We have got two major class of vaccines being rolled out for COVID-19. We have got the genetic vaccines, which are further divided into mRNA and viral vector-based vaccines. And then we do have some conventional vaccines rolled out in parts of the world. So one of the example of conventional vaccine is a SRPharmS inactivated whole virus vaccine and a Sinovac vaccine. Both are from China. They slightly differ in the variation of the COVID strain they use to propagate and prepare the vaccine. But in terms of the formulation design, they are pretty much similar to each other. The third important vaccine, which is again whole inactivated viral vaccine, is a Covaxin from India. And they used a combined adjuvant along with the inactivated whole virus. One of the adjuvant they use is quite interesting, TLR7/8 agonist, which activates the B cells, and antibody response in these kind of adjuvation is a lot better than the LM-based adjuvation. Another inactivated vaccine, which is still going through phase 3, some early results we have seen looks very promising, is a French company called Valneva. It's also an inactivated whole virus vaccine. Again, you've got TLR9 agonist, which has also been used in Hep B before. It's a very good adjuvant. Again, it stimulates B cells and produce more antibodies in response to inactivated virus. Another inactivated vaccine is from Medicago, which is spike protein based. Again, it's a subunit protein vaccine adjuvanted by AS303 from GSK. This particular adjuvant had some checkered past from the previous pandemic in swine flu with the controversy around pandemics. This has not gone through phase 3. Results are not quite promising at the moment, but they've been partnering with Sanofi and Clover Biopharmaceuticals to see if they've got other formulation variants which could perform better. Novavax is another subunit spike protein which has been engineered by biotechnology in an in vitro environment, and it's a nanoparticulate-based vaccine, which is again a nanoparticulate-based viral-like particle produced from the spike protein, which has been adjuvanted by their proprietary Matrix app. It's been produced on a plant cell line compared to other vaccines which uses animal cell line, and the efficacy trial has also been done in UK, and results are very promising, particularly against some of the troublesome variants in the world. It's not been approved yet. It's under regulatory review. So in terms of the mechanisms, how those genetic vaccines work, again, you all know about that, but just try to catch up on it a bit, because some of this would relate to how the clots are being formed, probably. So the mRNA vaccine, we use the messenger RNA, which encodes the spike protein, and we encapsulate this in a lipid capsid, and that's been injected into the body. Spike proteins are being then produced by transfection of these genetic code inside the muscle cells. These spike proteins are then released out of the cells. Some are then presented on the cell surface, which is being recognized by the immune system. You get the cellular immunity, you get the antibody response. A couple of examples are Moderna and Pfizer vaccine. It's very important to see this through to understand how potentially then clots may have been formed. The viral vector vaccines are slightly different. You encode the spike protein gene within the DNA of the adenoviral vector. The adenoviral vectors are silenced, so they do not have ability to multiply once they are in the host cells, but they are active viruses, and they transfect the cells, infect the cells, and transfer the genetic code. The spike proteins are produced again as a response, and then you get a lot more cellular immunity and a lot more antibodies response, humoral immunity as well. The examples of these kind of delivery system, we have got AstraZeneca, CanSino Bio, Johnson & Johnson or Janssen vaccine, and Sputnik 5 from Russia. AstraZeneca uses a vector which is chimp-based. It's again a silence vector, just to make sure that the pre-existing immunity against the adenoviral vector doesn't affect the vaccine efficacy. It's been seen in the previous studies that the pre-existing immunity against the adenoviral vector causes problems where the immunity starts attacking the vaccine particles itself, and you lose the efficacy. So AstraZeneca used a chimp-based vector to ensure that you get a good immune response. CanSino Bio from China used AD5, again it's a silence vector. Janssen also used AD26, slightly different variation. It's also a silence vector, and it's been used for a different vaccine production in the past. Sputnik 5 uses combination of viral vectors. The first dose use AD26 and the second dose use AD5 to make sure that after the first dose, the immunity against the viral vector doesn't affect the efficacy of the second dose. So they use a 2 different vector system to overcome that problem. The other companies has gone away with the first dose because the second dose would potentially then reduce the efficacy and have an immune response against the vaccine. It's the problem we see with the AstraZeneca trials last year with the half dose and double dose kind of issues coming up. Just a disclaimer, lots of confusion these days. People are trying to compare the efficacy of the vaccines, and people looking at the data saying this vaccine is better than that one, this vaccine is more efficacious than that one. And we raised our concerns back in November 2020 when Pfizer advertised 95% efficacy. We were concerned that these over advertisement of efficacy might be a problem later, and now we see the problem. Lots of people are demanding a particular brand because they feel that's more efficacious. All these efficacy numbers are arbitrary. They're not comparable to each other. The trials were not designed. They were not conducted in the same time period. The virus was mutating. It was in different geographies. And the trial design in terms of working out efficacy calculations and algorithms are also very different. The formulas used to estimate the relative risks are different. So we should never be comparing those together. In terms of the adverse drug reactions, this is the picture we found in our pharmacovigilance analysis with the MHRA data. So MHRA looks at the data within the United Kingdom, and this represents the cases represented from January to March. And this includes the Pfizer vaccine used within the country in United Kingdom and Oxford Astrazeneca vaccine. When I and my team was looking at this adverse events coming out from the database, it was very difficult to look at the signal in the beginning, but we found that if we go deeper and analyze the data in a different format, we could pick out quite a lot of clotting system events early in the March. At that time, the problem was not very much highlighted. There were a couple of cases reported in the Europe. Germany was insisting there might be a causal link, but MHRA did not detect the early signal at that time. But when we looked at the data, we could see there were strong signals. We could see thrombocytopenia, we could see the bleeding events, we could see the thrombotic events, and the total defects were too high, and there were fatal defects as well. And later, when the data analysis was more open in the public, then we did realize that the MHRA did miss the early signals. So signal detection in pharmacovigilance is not an easy job. It's a difficult work. You get thousands of reports, thousands of symptoms reported, and try to spin those together to find enough sensitivity signals against the background. It's just not very easy. Often adverse events are reported in different categories. For example, at that time, the issue was only with the CVSTs, but lots of clinicians were reporting cerebral thrombosis or thrombosis as a general category. So when the events come through from the hospitals, they don't sometimes specify where the thrombi is being formed. So if you're just using a keyword search, you will only find 3 events, but you need to go deeper, try to see what the coagulation system events are. Is there any link between bleeding, thrombocytopenia, and clots being formed in different anatomical locations, which are slightly strange? But often this requires thousands of telephone calls back to the hospitals, to the doctors, find more information, and pharmacovigilance doesn't work like that. They often look at the statistics, look at the background, try to detect signals, and when they have got strong signals, they try to then investigate it further. If you get lots of false positive signals early on, you spend a lot of resources to investigate a lot of unrelated events, but that would be more safer approach, but more intense, risk intense approach. So lots of regulatory agencies have put their algorithms to detect signals in different thresholds, and that's where you find differences in how the events are being investigated. In some regulatory agencies, any severe event leading to death or potentially fatal event, even if it's 1 in millions, is investigated thoroughly, irrespective of the frequency. In some, they rely on signal to noise ratio. Recently, on 22nd of April, just a few days ago, MHRA has then put in another update, and now they've analyzed the data slightly differently. They've put together all the events, they've investigated the cases more, and they've tried to find out how many thrombotic events had concurrent thrombocytopenia in there. And we can see the numbers were doubled in 2 weeks. We got 168 cases for the clotting events, major clotting events, which are life-threatening and fatal events, which were concurrent with thrombocytopenia. And only CVST was at least 77 cases. So it's just the way the investigations are done. Sometimes the signals are being detected, sometimes they're missed. So we looked at what could be a potential reason for thrombocytopenia back in March. So this is something we proposed in 15th of March, so quite a while ago now. It was a likely mechanism which we thought, and we know that from our experience that RNA viruses always infect platelets directly or megakaryocytes, the precursor cells. And in the past, we have found problems with H. pylori, H3N2, HIV, and more importantly, from Pakistani context, the dengue virus. We have seen that these viruses, mRNA viruses, do infect and attract an immune destruction of platelets, and messenger RNA has been detected inside the platelets for these viruses. So we thought maybe there is a similar story there, and these maybe viral mRNA are from the vaccine are being able to transfuse within the platelets. And platelets are very good in translating mRNA into proteins, are very good in cooking proteins. and that might be attracting an immune response against platelets or megakaryocytes, which might explain the depletion of platelets in these individuals, in vaccinated individuals. We have seen, if you look at the vaccine data, we have seen immune thrombocytopenia with flu vaccine. We've seen it with poliomyelitis, pneumococcal hepatitis, MMR, rabies. It's a minor effect. It's not very common. It's again a rare effect, but we've seen it in the other vaccine. It's been documented in the vaccine information documents from the regulatory agencies. Then there was another report just 2 days later from Germany where they identified an antibody in those individuals against PF4. And that was a breakthrough in terms of how people were trying to define vaccine-induced prothrombotic immune thrombocytopenia, and that started to become a criteria in a lot of clinical settings to define causative relationship with the vaccine. Now, this was a step change, but then later investigation found that PF4 antibody is not necessarily be found in every individual, and there were many cases then further came out where thrombocytopenia was there, thrombosis was there, it was very fatal, but there was no PF antibodies formed. So that's one manifestation. Some people do produce PF4 antibodies, and it's important to check, because then the clinicians in the front line need to know if they can use low molecular weight heparin or not. If there are anti-heparin antibodies, obviously then your course of intervention is different. So it's still valid to do the screening of any cases you're receiving in the hospital. The same research group from Germany then came back on 20th of April, just a few days ago, and tried to look at the further mechanism, how they could explain PF4 antibodies being formed. And they proposed maybe some of the vaccine excipients might be responsible for triggering this cascade. And one of the excipient they identified was EDTA, and they believed it might be causing in microvascular leakage. And they present some nice data. They did some experimentation and tried to put this hypothesis up. It's not been proven yet. It's still in the preprint stage, so it's not been peer reviewed, but that's the mechanism they come up with. In my personal opinion, when I look at that, I feel this might not be the causative link, because EDTA quantities in the vaccine is very, very tiny. And some of the vaccines have also got similar excipients being used in the past. It's in micrograms per milliliter, half ml of the dose, so it's further down half. Plus, in laboratory setting, EDTA is a chelating agent, which is very good anticoagulant instead of coagulation cascade producer. So still, we don't know. There might be a causative link, but it's still required for the investigation. Then we looked at the other possibilities, and we went into the regulatory dossier assessment report. On 2nd of April, we come up with a couple of studies which has been done in rats by Pfizer to investigate the preclinical safety and toxicity of the vaccine candidate in animal models. And what we found that the study 38166, which was done with the Pfizer BNT162b2 candidate, they found a strong increase in acute phase proteins post-vaccination. And that was linked to the acute phase reaction. This was expected, but we particularly could see that you have got fibrinogen elevation, and you could see macroglobulin increase and A1 glycoprotein increase, which all three of them are very important kind of element of coagulation cascade, and they are all prothrombotic indicator found in the animal studies of the vaccine. So this is the dossier submitted for the vaccine registration to the MHRA and EMA, and it reports that because loss of animals in those studies would not end up with the pharmacodynamic endpoints. For example, they would not form clots, they would not have CVSTs, so there were no safety signals raised at that time. But if you go back and reflect on the data, we could see that in the preclinical studies that there were some hints there. If you look at the study 513351 in mice done for the COVID vaccine Astrazeneca, we can see same acute phase response. They do not specify which proteins were increased in the report, but they do mention that the same acute phase response was seen. So we would expect, factor 1 macroglobulin and glycoprotein increase. But they also report that there is a change in plasma globulin to albumin ratio, which is again causes changes in the ESR, and the viscosity of the blood would lead to more thrombotic state. Interestingly, we also looked at the link with the PF4 antibodies, and we found that some of the acute phase reactant proteins are also heparin binding proteins. And that's interesting, because if you've got acute phase reactant proteins are increasing and some of them are HBPs, then it's expected that the immune system might react against that and produce PF4 antibodies. So although the patient had no exposure to heparin before, but for that reason, you could explain why indirectly PF4 antibodies were raising in some of the patients. But again, there's a huge intersubject variability. Not everybody would respond in a similar way. There is a predisposition in some people with some pre-existing coagulation disorders, some people with a history of taking chronic anticoagulants or past thrombocytopenias, or people with maybe other genetic disorders, which put them on more predisposition to thrombotic risks. So what we did further on April was we were trying to explain further. We thought the mechanism is quite generic. We should be expecting similar behavior from mRNA vaccine and vector vaccines, but the pharmacovigilance data was saying that Astrazeneca was hitting on the brain more compared to the Pfizer Moderna vaccine, where the clot and thrombus formation was all across the body, but less in the brain. And we wanted to see more as to how we can explain that peculiarity between the two types of technologies. And when we looked at the dose, we can see that the dose per injection is quite a lot. So you're injecting about 50 billion particles. In case of Oxford and J&J, it's about 100 billion for Sputnik 5. The question was that if you inject the vaccine in the muscles, would that stay in the muscle? Is there a targeted approach used in the formulation design, which would keep it combined and to the muscle region only? And we found it wasn't. And we again went back to the biodistribution data from animal trials, and we found the study 841MV, which was not done with SARS-CoV-2 encoding vector, but this was a different viral vector, HBV, so a different virus, hepatitis B virus vector, encoding gene, but the same chimp ADOX1 vector. And when we looked at that, we could see that the vaccine is moving away from the muscle site. Within 24 hours, the adenoviral vector encoding the HBV virus gene was found in lots of other body tissues, including brain, which made us a hint, thinking about would that explain why some of the clots are particularly formed in the brain with this particular viral vector vaccine. So there was another study, 514559, which looked at the biodistribution of ChAdOx1 encoding particularly COVID gene, but that study wasn't published at the time of the regulatory approval. This study was still ongoing; it wasn't completed, and we haven't got the access to that data yet. But that study could shed further light about the biodistribution kinetics of the vaccine, and if this could explain some of the adverse reactions we are seeing. But the same vector encoding a different viral gene, that's the indirect, evidence we can see that could explain. When we looked at the Pfizer vaccine and we tried to find the biodistribution data, we found that, again, it wasn't performed, but there were some surrogate studies done with luciferase and same lipid capsid, same, the formulation style. And we could see that after injection site, the vaccine traveled down to the liver and the body tissue. So it was also moving away from there. Same story for the Moderna, encoding, cytomegalovirus. It's again, the vaccine for COVID-19 was not tested, but there was a similar platform used with a different virus. And we could see that the lipid nanoparticulates taking the viral mRNA were going beyond the injection site, particularly in the eye and spleen as well. So we were trying to find out why those studies were not completed before the emergency use authorizations were done, and we found that they were not required by the regulatory bodies, because traditionally vaccines are considered as an immune response therapies which do not work like a pharmacological classic approach, and they work by an immune stimulation system. So this study particularly wasn't required. But that may be true for conventional vaccines where we inject inactivated viral particles or maybe use subunits. But these mRNA or viral vector-based vaccines are gene therapy approaches, and the biodistribution is very important to see where the transgening vectors or the lipid carriers are going away from the vector side, injection side. And that would explain quite a lot of information when we come to the ADRs and tolerability in some patients. That's a very interesting study we found back in 2007, so a long time ago. And this is, again, one of the adenoviral vector. And we know from the past that these adenoviruses are hepatogens. They always sequester in the liver. So using these vector, adenoviruses as a vector, there's a strong possibility that they will activate platelets directly and will sequester in the liver directly. So in this particular study, in animal models, they could particularly see that after intravenous administration, 85 bound to circulating platelets and caused activation of platelets directly by the viral vector, and then it was sequestered in the liver and been destroyed by the Kupffer cells. And that could explain, why these viral vector vaccines may be more attracting a thrombocytopenic response after injection in many subjects. This is information coming from American Society for Hematology, and you all are aware of that. I don't need to explain that to you. You already work in the front line. You all are doctors, so we know that from our COVID experience that one of the key reasons why coronavirus disease caused so much case fatality ratio, so many people died. There were two main reasons we learned last year. One was a cytokine release syndrome, a very hyper-inflammatory state, organ failure. And the second we learned was the COVID-associated coagulopathy. And we all know that coronavirus was causing a hyperthrombotic state. And we know that the hospitalized patient has got very high fibrinogen and D-dimer, got prolongation of PTA, PTT, and we see thrombocytopenia in patients as well. And when the patient progresses further to a severe stage and a multiple organ failure starts to kick in, we can now see a very different stage because fibrinogen goes down now, because now fibrinogen has been activated to fibrin in most of the cases. So actual fibrinogen is now very less. And that's interesting because now on one side, the D-dimer is going very high, but fibrinogen is going really down. But you go further prolongation of PT and APTT, and thrombocytopenia is getting worse. And that's a similar pattern which you could see in treating sepsis, in DIC. And taking that knowledge, we know that that's done by the H1 virus. And in the vaccines, we are using the same components of the virus. So we were again trying to link what's the correlation there, because the virus caused that, and then puts patients, severely ill patient, into that hypercoagulation state. Coagulopathy is one of the cause for fatalities. And how does that explain? So we went back to the, again, spike protein theory there, and we looked at the spike protein, and one of the component of the spike protein, which is a receptor binding domain, this green bit on the top. This bit is important because that's what binds with the ACE2 receptors and causes lots of pathologies associated with the virus. So there was an interesting data we found from the phase 1 clinical trials of Pfizer vaccine in China, and that was BNT162B1 candidate. So the current vaccine, before you look at that data, the current vaccine in use in US and UK is not that vaccine. That's a B2 candidate. The key difference between B1 and B2 candidate is that the B1 candidate encodes an mRNA which only translate receptor binding domain. So that bit of the spike protein. The B2 candidate encodes the genetic code mRNA for the whole spike protein. So when the body would translate that mRNA, you get the whole spike protein being produced, which is the current vaccine we use. But that vaccine only used the RBD domain on the tip of the spike protein. So in the early stage, the Moderna BioNTech had got a few candidates, and they trial the safety, antigenicity, and tolerability, particularly in animals, and phase 1 human volunteers. And based on the data, they picked the best candidate for further development. So this was dumped after the phase 1 human trials. We could see a very acute phase reaction. You can see CRPs. You can see here leukocytes in day 1. You can see the lymphocytes going down in day 1. Now, these numbers are interesting because you have only next measurement done after 1 week. So you don't know if this goes further up, and then it comes down, or that's the true maximum, you could say. And that's not known because in the design, they were only testing it after 7 days. But lots of this hematology data and clinical biochemistry data is not released in phase 1 and 2 trials. Most of those trials only shows the pharmacodynamic endpoint if these changes resulted into any adverse reaction, any thrombosis, hemorrhage, or any kind of thing. If nothing is being reported, no severe adverse reactions is being reported, then phase 1 and 2 data would not show that much detail. But that particular candidate, luckily we found some details, which was published in a preprint server, and we could see that interesting behavior. And the more important thing here is the platelets. You can see it causes thrombocytopenia on day 1 after injection. And again, that's not a true minimum. We don't know how far it goes down before it comes back up. These volunteers were perfectly healthy volunteers, no pre-existing conditions, a very long exhaustive list of exclusion criteria, because this is phase 1. You won't want to risk it in a troubled volunteer. So majority of people reverse, acute phase lasts for a few days, and then the hematology markers come back to normal, which is why you don't see adverse reaction in majority of people when this vaccine is rolled out in mass scale. But there are some people who react to that phase, who can't come out of this, and they produce adverse reactions before they kind of stabilize their markers. So there's a link down there for the full study, and data is available publicly. It's nothing confidential here. So then we looked at this spike protein as a explainer, and we thought maybe this spike protein is responsible for the coagulopathies. Maybe this is the reason why the virus is interacting with the platelets. And the receptor binding domain is the key, because a small molecule, the molecular weight is low, the size is tiny, and its distribution pharmacokinetics is a lot more deeper compared to the rate constants you could calculate for the bigger molecule of spike protein, which will entrap here and there, would not be able to go through the leaky junctions. So wherever the ACE receptors are in the body, RBD domain might sneak in and bind to lots of other ACE2 receptors, cause a lot of safety issues. So that candidate was dumped. The whole spike protein was safer candidate. It was a bigger molecule, doesn't go around much, stays in circulation, and then body clears it out. But during circulation, it interacts also with the platelets, and that might explain why lots of thrombocytopenia associated with even B2 candidate. So then we looked at back again to some more evidence generated during COVID time. So this is again 2020, trying to understand how virus is entering into the body. This was the first reference in Lancet, which explained, how the knowledge about these 2 receptors, and particularly these transmembrane protease, TMRSS2, conditions the spike proteins from the pre-fusion state to the post-fusion state, which allows the virus to enter into the cell. And that was very important because these 2 parameters were the key in mediating the virus entry and pathogenesis. Now, that's a very important information if you keep that in mind, because the way the virus is doing this is through these spike proteins and the receptor binding domain, right? And if that spike protein is being produced by the vaccine, or vaccine is being using those spike protein as an antigenic target, then those spike proteins, if they're not contained in the muscle site, if they've been going out in the blood circulation, then they will be interacting with these receptors down there as well and causing similar effect. Now, this was, again, 4th September 2020, old reference before vaccination. And they found that the SARS-CoV-2 binds to the platelets ACE2 receptors. and causes thrombosis. So when people were trying to understand why so prothrombic disease, why the virus is causing so much coagulopathies, then they found that there are ACE receptors on the platelets, which are then activated by the spike protein directly by the virus. And again, TMRSS, a transmembrane protease, is also being expressed on the platelets, which provides the best environment for the spike protein to then bind into the platelets, infect them, immune system attacks, and then you got the thrombocytopenia. And that's the likely explanation we see, and that's the evidence goes down and explains how the spike protein does it, whether the spike protein is part of the H1 virus or the spike protein has been produced on the cell surface, which has been spared by the vaccine to initiate the cascade. Linking that with the new Oxford study, you might have all seen, where they've tried to work out the risk of thrombosis from the H1 virus compared to the vaccine. And you could see, there's a very high risk they worked out for the COVID-19 itself. So they were trying to say, look, for the CVSTs to be developed from the H1 infection is a lot higher than the H1 vaccine. So the vaccine is still, kind of risks, outweighs, the benefits outweighs the risk kind of theory there. But bear in mind that this paper is not yet peer reviewed, and there is an important consideration in the data. So if you look at the data, this is a relative risk. This is a statistical calculation of the risk of getting CVSTs. This is not the actual data that how many CVSTs were found in how many positive patients. So that's not an absolute count. It's a relative risk. So it's a hypothetical, theoretical risk being calculated to work out how much chances I have got if I've got the disease. How much is the chance that I would going to get CVST? Also, they have not used CVST in particular. They've kept the broader term to make sure that they're capturing the maximum risk associated with the virus. For the mRNA vaccine, you can see they've shown a very comparable behavior with the Oxford as 5 and mRNA is 4. But the tricky bit here is that the Oxford data comes from the EMA, which is the absolute numbers. So how many cases were found compared to how many injections, and that's not a risk. And the mRNA data comes from relative risk calculations. It's not a head-to-head direct comparison. And if you look in the brackets, you can see the confidence interval is as low as 1.1. So the risk could be 1.1, not the average value of 4.1 in majority of patients of vaccinated people. So it's not a good comparison, to be honest. And same issues with the portal vein thrombosis. And when they extended the definition further and look at the CVTs, there's no comparison with Astrazeneca vaccine here. So when you see these studies, making headlines in the press, when you dig down deeper into the science, then you can find more correlation and better understanding of how you could correlate it. So there's a caution there. What you see in the press, what comes out has to go through vigorous peer review and scientific discussion to then discuss the pros and cons of the theory being explained there. A couple of interesting case studies I would like to point out is one of them is in the Lancet for a 50-year-old woman. And you can see pulmonary embolism, thrombocytopenia, nothing in the brain, Oxford vaccine, no PF4 antibodies, but the thrombi was formed in the pelvis, went up to the lungs and caused the embolism. Very important that you not necessarily you're going to find PF4. And it's very good case, very short case. There's 2 minutes explainer video down on the Lancet website as well. I would recommend all clinicians watch that, and they explain all the diagnostic markers they've used and how they treated the patient, and patient didn't die. The another one was this one, ophthalmic vein thrombosis, again, very different, ischemic stroke, and again, concurrent immune thrombocytopenia. So have a look at that one. The next one is very interesting case coming from India, just came on 14th of April. And that was the Astrazeneca, Indian version of the Astrazeneca. And they found that the clot was formed in the coronary arteries and caused the MI. And that opens another door that lots of pharmacovigilance databases are coding myocardial infections and heart attack and coronary events separately. And they've never been looked at into thrombotic events to analyze. And we don't know how many myocardial infections, hundreds of MI and heart attacks being reported in the PV database. We don't know how many were actually clot mediated? How many of them were concurrent thrombocytopenic as well? We don't know. And that requires a lot of hard work for the regulatory bodies to investigate every single case. So the official advisory for all the healthcare visitors for the European Medical Agency is that thrombocytopenia is now officially recognized as 1 in 100 or even 1 in 10 event after vaccination. So it's going to be very common. And because it's so common that majority of healthy would be fine, but there are minority of people with pre-existing condition who might, come up with adverse reactions. So you need to bring that in mind, that the usual symptoms for stroke and kind of these fatal clots in the brain, and make sure you educate the patients after vaccination that if they've got any of these kind of symptoms, you bring them early on. CVST can be life-threatening, but if you intervene early on, if the patient is not ignoring the symptoms for a whole week, then we can save them. There is a treatment now. Lots of case studies are being published in Lancet, where the clinicians can then guide themselves how to treat those things. But more education we would put in, less fatalities we would found, less people dying after the vaccine would going to help us with the vaccine uptake eventually, because that balances it out. So that would lead us to more risk group being recognized now by the MHRA in UK. So the official advisory includes women with contraceptive pills. So when we looked at the Germany case studies, we found majority of women who were reported to cause these CVSTs in the early stage have got a concurrent use of contraceptive pills as well. So if somebody is on contraceptive pills and get the Astrazeneca jab, there is more likely that they might end up with these issues. If any previous history of heparin-induced thrombocytopenia, then that's a very big risk factor. If people would have got any clotting abnormalities anywhere in the body, or they reported thrombocytopenia after the first dose, again, they shouldn't be getting the second dose. And previous incidences of thrombus with other vaccines, if they've got another problem with another vaccine, then they shouldn't be giving that one. History of immune thrombocytopenia as well. Thrombophilia is not officially recognized yet. There is not much data out there. So this is not in the official advisory, but I think that should be a cautious group as well, because they would have a genetic predisposition to form clots anyway. So if you induce thrombocytopenia in those people, they would gonna react. And those population who are on chronic anticoagulants, again, you need to watch out, make sure you check their coagulation profile before they've been given vaccination. Another early indication, a signal we detected on 18th of April was the heavy menstrual bleeding. And that's been reported by thousands of women on social media platforms. But when we looked at the official database, there are only 958 cases reported in our MHRA database. There are similar events, almost similar numbers in US. The numbers are less likely to be reported because lots of women wouldn't report it because of the cultural context. Maybe they think it's not vaccine related, or maybe they tried to report it and they were not encouraged by the clinicians because they thought it's not related. So there are lots of underreporting there. And we've found that this heavy menstrual bleeding may be explained by the thrombocytopenia. And because dangerous CVTs are also more prevalent in females, that explains, there might be a link. And the significant loss of blood or hemorrhage, we've seen loss of vaginal hemorrhage and uterus hemorrhage being reported in pharmacovigilance databases, could lead to severe anemia, further exacerbate the thrombocytopenia and increase the risk. So the bottom line is that if women are complaining about this, we need to flag it up, make sure we report it, make sure we check it, make sure we give them a coagulation profile, make sure we check their thrombocytes to prevent, they progressing into a severe condition. Do not just ignore it, because we think it's not related. So reporting PV numbers are very important for any new medication. Even in a clinician judgment, if we feel it's not related, it's our duty to report it. Because say if an unrelated event is reported by 10,000 people across the world, then it might create a strong signal for MHRA to investigate it further. But one case you might not bother investigating. So if you do not report it, the data is not collected. If the data is not collected, there's nobody going to put in investigative signs on it. So make sure you report it. If you feel it's not related, you can write in your narrative So in the reporting system, there is a paragraph, there's a box to write about the clinical judgment. So you can use your clinical judgment, make sure you provide the age, make sure you provide the batch number of the vaccine, provide as much as information as you can. Because now when we look at the endpoint, we can't find information. We don't know what the medication patient was using. We don't know the medical history. And we need to then go back and trace the case, call people, call physicians, call doctors. And that takes a lot of time, particularly when you got thousands of reports coming on a daily basis. I can also think of some other potential risk groups. Again, these are not officially recognized, but people with immune compromise. Remember that once the vaccine has infected the body's healthy cells, then the spike proteins have been produced, the cell becomes defected, and that defected cell needs to be cleared off by a very good T cell response. So if somebody is on immune suppression, if somebody is taking T lymphocyte antibodies, then you might expect that that person might struggle clearing this off. So that's another risk group, HIV, hyperimmune. So people talk about efficacy of the vaccine in those because they think they're immunocompromised, so they might not produce good immune response. Efficacy is one issue. You can live with a poor efficacy, but safety is also important, which I'm trying to point out here, that once the vaccine has transfected the body's cells, and particularly when the cells are not just muscles, it's going away from the muscle side and other part of the body tissues, how are you going to clear up if the T cell response is not good? Again, people with severe allergies, so you try to prevent anaphylactic kind of reaction. People with active cancers, where we know that the body's response to remove the defected cells is being hampered. Just a very brief shot, if I've got a few more minutes, to give you a picture of Pfizer mRNA vaccine. So we're also looking at some signals of the Pfizer's mRNA vaccine, and we can see some strong signals, which has not been yet linked to the cause, but we can see some very strong signals, which could hint that there is still a correlation there if we investigate these cases further on. So if this is the US data only and only Pfizer vaccine, and we can see there are 322 major thrombotic events, although CVST is only 3, but the DVT is 135, and there are about 15 pulmonary embolism or clots, and 155 further thrombosis being formed. We don't know how many of those were accompanied by thrombocytopenia, which is a bit of a problem because we don't have that level of access which the regulatory bodies have. But these early signals has to be investigated further to then find out what's happening with the other similar vaccine. So in terms of bleeding events, about more than 400, hemorrhages, particularly in cerebral hemorrhages. But early signals are very important. Nosebleeds, eye bleeds, gum bleeds, the heavy periods. Hundreds of those cases, which are very good sign that if somebody's coming back with a nosebleed saying, I've never had nosebleed before, had the vaccine 3 days ago, got nosebleed, not stopping, get a thrombotic check, offer a coagulation profile, check the platelets. Maybe he's suffering from an early phase response and have got thrombocytopenia. Majority will recover in 8 to 9 days, 10 days, people will recover. Minority might end up with a severe effect later. So it's good to be vigilant and pick on those minority of people who might be at a high risk. Again, if you look at the stroke, now stroke is another big signal there. Now, in the initial report, sometimes clinicians don't explain all the details in the form they fill in. And we know from our knowledge that, majority of strokes could be mediated by a thrombi, in the brain. So we don't know how many of those are, but we can see lots of CVAs, lots of cerebral hemorrhage with the vaccine, and lots of cerebral hemorrhage seizures reported as well. Combining this with thrombocytopenia, again, you've got about 76 cases of thrombocytopenia reported. But again, the problem is some cases are reported as a low thrombocyte. Some cases are reported as low platelets counts, and different keywords have been used. So I'm not even seeing the full picture here because I've not used all the possible keyword search to get the full data out. But there are lots of other events reported by different terminologies, which needs to be grouped together and then analyzed properly. But there are at least 76 thrombocytopenias there. This is the situation with potential deaths associated with the reported after the vaccine, if you like. There's more than 1,000 cases there, which might require some more investigation. So pharmacovigilance is not an easy job. You get thousands of reports, you got signal detection, there are algorithms and computer programs to detect signals, which send safety messages, which then goes to telephone calls, call centers, investigations, patients, clinicians, coming back to data. It's a big cycle. It's not easy. Some regulatory bodies have got very sensitive signals management system. They detect it early, some detect it late, but clinicians are at the core of this system. So as a clinicians, if you report everything in a lot more detail, if you provide a lot more investigation, if you check the platelets, if you look at the history, if you look at the concurrent conditions, if you look at the past condition, it helps regulatory bodies then to evaluate the full risk. Just to put it back to the perspective, there is a controversy with pandemics about 8-9 years ago in the swine flu pandemic, and after 8 years we begin to realize that there are some signals missed, and there's a second lawsuit now just filed in United Kingdom in 2020 claiming the link with narcolepsy with the swine flu vaccine. And this has never been recognized. The causal link was never been established, but the data presented to the court proceedings in the first case shows a very clear difference now after 8 years for a particular formulation or brand of the vaccination compared to the other products which were out there at that time from the United Kingdom. It's only the UK data at the time presented to the courts. This is publicly available on that BMJ website. You can have a link. Just trying to put it into perspective, saying, there is lessons to be learned, and we have to be vigilant. Vigilance save lives, even if the effect is in a minority, even if only few people are going to be affected. If we could save them, then we could overcome vaccine hesitancy a lot better. If people are not going to die with the vaccine will be perceived a lot more safer, and we don't have to really do a lot more hard work to explain people, that why those events are happening. So it's all contribution from different professionals, and healthcare professionals in the front end have got the biggest responsibility, and they're so important to help us and regulatory bodies to inform the decisions. So that's where I would stop. Thank you very much. If there are any questions, I'm happy to take them now.
Thank you very much, Dr. Hamid Merchant. It's a wonderful presentation and very in-depth knowledge and analysis. So I will be coordinating some of the questions which were asked during the session. The first one is Mr. Tanveer Ahmed. Please guide us. Sputnik vaccine is approved by FDA or European Drug Authority or any stringent regulatory authority?
From last night, the EMA has started looking at Sputnik data. So they've confirmed in a press release that they have now started looking at the data, but they mentioned that they're only on a very early stage. And they said because they're now aware of the viral vector vaccine causing these minority of events, so they are intending to look at it in a more finer tooth comb to make sure that they're not missing out anything. But it's a very early stage of the review. However, we know that some independent member of states in the EU, they have given EU emergency use authorization in Europe, and vaccine has already been started to be used in Europe in some places. But EMA has not granted a emergency use authorization as yet. Same situation with the UK or the US.
Okay. So the next question is Dr. N.M. Bhatt. And their question is that what is the percentage of side effects, how severe these are, and what are the side effects of Chinese vaccine, particularly that is the CanSino. So basically, you know that the free access in Pakistan is Sinopharm, and now there is a CanSino Bio. So their question is the side effect profile of both the inactivated Sinopharm or Sinovac vaccine and the vector-derived CanSino Bio vaccine.
So the honest answer is that, we speak with the evidence, and because other vaccines are not being used in UK, Europe, or America, our pharmacovigilance systems cannot detect or monitor the adverse event profiles. So we don't see it, we are blinded with the data, and there's no open source monitoring system where we can assess the safety and efficacy of those vaccines. So it's very difficult to comment, but what I've tried to do is try to explain you some of the mechanism behind clotting, and try to link the science with the adverse event. And I've also tried to explain you the formulation of different vaccines. So for example, how CanSinoBio's viral vector vaccines is similar or different to the AstraZeneca vaccine. And that knowledge can be used to translate what would be the likelihood of causing similar effect. But if you do not collect data, if the pharmacovigilance systems in Pakistan and other countries are not very actively collecting information, then we will be blinded. Nobody can tell you what the adverse event rates are. We are in phase 4 post-marketing surveillance studies, and pharmacovigilance data then brings in the adverse event profile. And that's what we get in terms of what is minor, what is rare, what is common side effect. So all these numbers comes out of this phase, and every country has a responsibility to contribute to that exercise, otherwise we'll be blinded.
Thank you. Another question is from Azhar Hussain. Could we say that killed COVID-19 virus vaccines are relatively more safe than vector or genetically modified or messenger RNA?
So very, very good question. If you look at some of the mechanisms we have explained, if we look at the cause of the thrombotic events in the brain caused by the vector going to other parts of the body, viruses have got much more deeper distribution kinetics compared to the non-virus vectors. So if that's the causative reason, you might think maybe the viral vector vaccines are causing these minority severe events more than the inactivated virus vaccine. However, if you believe on the mechanism of the spike protein mediated pathogenesis, so the spike protein is binding in with the platelets and causing these coagulopathies, then spike proteins are present in inactivated virus vaccine as well, in the kill virus vaccine as well, and they should be also exhibiting similar thrombocytopenia. They would lead to fatal clots or not, we don't know, which is why I'm saying it's important that we collect the data. So maybe they're causing thrombocytopenia, but that is very transient, and thrombocytes don't fall down to 20 and 30. Body is very clever. You don't start seeing hemorrhage and bleeding until platelets are really down at the bottom. So say if you go 100, 80, 60, majority of people would still not show severe adverse reactions. And if it's only a transient phase, people will pass through it. So we need to collect the data and see, but by looking at the information we have so far, it looks like inactivated viral vaccine are safer than the genetic vaccines. But again, we can't say it with full confidence because the data is limited.
Thank you. Another question, and as you know that the inactivated vaccines are not available in North America. So another question is Professor Nigat Mir Ahmed, and she is a rheumatologist, and she is concerned about the patient with rheumatic disease, particularly systemic lupus erythematosus and APLS. Her question is that should I suggest vaccination of public? Should it be okay with by the physician? So what should be her professional advice based on your scientific knowledge?
It's just difficult, to give a direct medical advice for a particular situation without looking at it in more detail. My advice would be to see every patient differently. Do not generalize it for a category of patient. But if somebody would have got a condition where they are being using immunosuppression, if you have already prescribed disease modifying agents to those patients, and if their immune system is already suppressed, some arthritis patients in UK are on very selective T lymphocyte antibodies as well, like rituximab, and we know that would shut down the T cell function. So you need to consider what is the patient, what's being given to the patient, and sometimes the pharmacokinetic half-lives are 6 months. So you can't just wait for a week and then give a vaccine. Maybe you might have been waiting for 6 months for T lymphocytes to come back. But look at it, a case-to-case basis, and also do a coagulation profile at the same time to check if the patient could bear the coagulation effects or not.
Okay, thank you. Another question is from Muhammad Siddiq. Their question is that most of the vaccine are having the emergency use authorization. So whatever the vaccine which have received the emergency use authorization, are they going to full registration by the FDA? And one that's probably going to happen.
Normally, in a pandemic situation, EAU is the legal way to get a fast track, and that's been done based on the risk versus benefit ratio, the need in the society kind of thing. But the idea is that the data is being completed. The studies will continue collecting more information, and it might lead to a full authorization. But if the pandemic is over and vaccine is not going to be used further, then there might not be full registration. I give you an example. In 2009, when we had the pandemics, the swine flu, there were 4 vaccines specifically being designed and produced for the swine flu pandemic. Those vaccines were given emergency use authorization, used throughout the pandemic, 4 different products across the world. And after the pandemic was over, those vaccines were withdrawn from the market. We never seen them before again. But the annual flu vaccination cycle then included some of the variations of the flu virus, which would give protection against H3N2. And that was a full, complete regulatory approval cycle was given for those, and they were in the annual immunization schedule. But the one which was produced in an emergency only for that particular strain of swine flu never got long-term use authorization. So it's likely that some of these vaccines will die after pandemic. They would not get a full authorization, but there might be an annual booster vaccine, which has a slightly different formulation. Because it's a different formulation, antigen is different. It will go through, again, clinical trials, and they might be given a full regulatory review and approval, because we don't need them immediately now. They might be needed before the next booster cycle comes in.
Okay. Another question, and which is a very important question. Do we know what is the level of anti-spike antibodies post-vaccination, which are known to be protective from the quantitative purposes? Because lot of physician, lot of patient, lot of the general population, they are concerned, they have the antibody titer, and which, based on your experience from back home country in Pakistan, it is quite variable. So their concern is, what level should we say arbitrarily is protective enough really to consider it safe?
That's a very good question, I think, and it's very important to understand that how these correlations are being developed, and why we don't have a vaccine passport as yet in terms of antibody titers. So the key issue is that the different vaccine products have used different antigenic sites, and different vaccines are producing slight variation of antibodies after vaccinations. So, for example, if you presented a whole inactivated viral vaccine, some of the patients are developing antibodies against the spike protein, and they are also producing a neutralizing antibody titer, NAB antibodies. They're also producing antibodies against the nucleocapsid proteins. The genetic vaccines, which are encoding specifically for mRNA on the spike protein, the antibodies produced as a result are very specific to the spike protein. So the antibody tests we have got in pathology labs are variety. They differ in terms of how selective they are to pick a particular antibody. They are picking a cocktail of antibodies, IgGs, or they're very selective for a particular method. So a majority of tests I've seen either are non-selective or they are very specific to spike protein. Some labs are doing nucleocapsid proteins, which I'm aware of. So it's very difficult to understand. Some people have done nucleocapsid antibody tests and they found, oh, I've not got a response. I've got zero titer. Vaccine didn't work in me. The vaccine they got was an mRNA vaccine, and it should be making spike protein rather than the other ones. So wrong test. Other issue to understand is that majority of antibody test has also received emergency use authorization, and they've not gone through full validation as you would expect. So we don't know which methods are correlating with the science, which methods are producing the right antibody titers, and which antibody titers are are linked to the efficacy. If you don't have that validation, we can't say this titer is required for protection because we don't know. And there's a T cell response as well, and we're not testing for those. So if the T cell response is providing you long-term immunity against the variation of the virus, it means if you don't even have antibodies, or antibodies are finished after 6 months, but the T cell response is still alive, then you would get a variation virus. You would get test COVID positive, but you will not fall ill. You will not be hospitalized. You will not be dead, because your body will react much faster, produce new antibodies. So it's very difficult to say, which is why intentionally it's not being done. Because if any country would like to jump in and try to impose those kind of criteria, it would be very premature and would be misleading. So I think the time will tell us more. The research is happening. Some immunologists are working on this direction. They're already doing it, and they're trying to collect this data from different trials and studies and trying to correlate it with the vaccine efficacy and reinfection rates in different population data. So we might take a few months or maybe a year to fully learn the dynamics of antibodies.
So just a couple of more questions because we are running short of time. A very quick reply. Another question, do you suggest to know the platelet count before giving a jab? Is it advisable or no?
Officially, we don't do it in UK because we have to then give it through the NHS. So it's very difficult job for us. But in countries where the system is private, the CBC and those blood tests are not very expensive. In Pakistan, you can get a CBC for a few hundred rupees. It's not very expensive. So if a clinician think that that patient have got coexisting condition, risky, maybe let's do a check. So what's wrong? I would say yes. Do a coagulation profile, do a CBC, and see if your patient would tolerate it. Also, there are lots of convalescent patients which were asymptomatic COVID positive. They never know that they had the COVID-19 in their lifespan. So giving them an antibody test for SARS-CoV-2 is also important to make sure if they've got a pre-existing titer, then they might not need 2 doses. There's a new study from just published in Nature and Lancet, the 2 studies actually, and they've confirmed that after 1 vaccination, convalescent patient produce enough titer equal to 2 vaccinations. So you don't want to overload people with antigen in a short span of time. Again, remember, it's 100 billion viral particles in some vaccines, 50 billion viral particles in some vaccines. They're not replicating viruses, but they're antigens. Your immune system will storm against them. And if you inject them on a very short period of time, 3 times, for example, 1 natural infection, then 1 vaccine, then another vaccine, some patient will react. So take a safer approach. If you can offer a test, do offer tests. Evidence-based approach is always good.
Thank you. And before going to the panelist, we have Dr. Ubaidullah from Drug Regulatory Authority of Pakistan. So we would like to request Dr. Ubaidullah to share, he is from the registration, that what is the level of pharmacovigilance which is being monitored or advised by our regulatory authority in Pakistan.
Thank you very much, Uzair sir. And first, Dr. Hamid, very good presentation and excellent coverage of all the points regarding the vaccine from A to Z. Regarding the landscape, national landscape regarding the vaccine, although some of the observations, points have already been cleared by the Mr. Hamid Marjan, but just to grab the overall situation in Pakistan, by and large, if we see that none of the regulatory authority of the world has granted marketing authorization to any of the vaccine. Rather, every country has granted the emergency use authorization based on the risk-benefit ratio of that very vaccine, and same has been done in Pakistan as well. Second point is all the approvals have been granted either on the basis of the interim analysis of phase 3 trial or on the basis of some peer-reviewed journals publications as well. There was a question regarding the Sputnik vaccine. In fact, if we see right now around the globe, it is an era of the vaccine diplomacy. Countries are making their relations with other countries on the basis of the vaccine supply and vaccine approval, not on the basis of some very good scientific evidences. And the same is the scenario in this part of the world as well. Sputnik vaccine is a very good vaccine, and probably because it has been manufactured in Russia. So due to that might be that reason some countries, developed worlds, are not considering for the approval But as Dr. Hamid has already shared that some European countries have been granted. So the data of the Sputnik has been published in the Lancet, which is one of the peer-reviewed journal, and it is an effective vaccine. As far as the grant of the approval of some vaccines are concerned, I just want to share one example of the Ebola vaccine. Some time past, it was decided by the stringent regulatory authority to approve the Ebola vaccine when it was in phase 2 trial. It was even not switched over to phase 3 trial. So it is the decision of the regulatory authority to grant the registration or to reject the decision based on the risk-benefit ratio. And for that very reason, we are saying that we have granted the emergency use authorization to various vaccines. So by and large, if we see our national scenario, that we have granted emergency use authorization to 5 vaccines, and out of these 5 vaccines, 4 vaccines have been administered in Pakistan, including Sputnik, Sinopharm, CanSino has been started, and another 4th vaccine, Sinovac, will be in the immunization cycle from the next week. Astrazeneca vaccine, which is the vaccine with the clot formation component, is still not in the has not been used in the Pakistan. So by and large, if we see that inactivated vaccines, Sinopharm and Sinovac vaccines are the safer vaccines. So the safety profile of these vaccines are acceptable, are in acceptable limit. As far as the query of the Uzair Saab is concerned regarding the pharmacovigilance, fortunate enough that we have started the National Pharmacovigilance Center 3 years back. And with this National Pharmacovigilance Center, all the 4 components have been connected. Although this pharmacovigilance reporting, ADR reporting, is not up to that level as Dr. Hamid Merchant is saying of NHS, but we have started working and we have been receiving various signals of the various ADRs reporting. So by and large, the signals of the ADRs are of the same level, or the same side effects have been reported as have been shared at the time of the clinical trial phase 3. So right now we can't say there are some signals which are different from as received during that trial. So, but we are continuously in the phase of monitoring. Our government has developed 2 oversight committees for the ADR monitoring. One is Clinical Management Committee of the epidemiological point of view, which has been headed by Dr. Asad Ali from the AKUH, and another committee is for the emergency use authorization purpose, which clearly and oversee all the trials data and then recommend. By and large, if we see there are about 80 vaccines which have been, or 100 vaccines which they started working before for getting EUA, but out of these only 5 or 6 has been approved by the various regulatory authority in this part of the world. And if we overall see, so probably there are 12 vaccines which have been granted, or 10 vaccines which have been granted EUA by various regulatory authorities. So if I summarize, then the pharmacovigilance activity in Pakistan is not of very good shape, we can say, but is right now is in a position to detect the signal if there is something went different from as has been reported. And we are very much particular about the CanSino Bio as well, because it is the vaccine which has been granted EUA on the basis of evidence-based clinical trials conducted in Pakistan. So we are very much cognizant of the fact, because it is a vaccine which has been first approved in Pakistan and then other parts of the world. So we have planned various pharmacovigilance activities specifically for those vaccines which are being tested in Pakistan and which will be used in Pakistan, especially CanSino. As far as there was one query from the Sinopharm is mostly has been used in UAE. Probably it was the first vaccine which was approved by the UAE government, and UAE government has a very good pharmacovigilance system as well. So we can say that we are already working on the pharmacovigilance system, and in the coming week, when we will be using CanSino Bio, then we will be more vigilant for all the ADR reporting and all the things. Thank you.
Thank you very much, Dr. Ubaid. We have so many questions, but the time is definitely running short. Just a quick feedback from Dr. Umar Chuktai, because he was very close based on his observation, some special tips really from the diagnostic purposes and, from the immunization. Just a couple of sentences, please.
First of all, thank you so much, Dr. Uzair, and thank you so much, Dr. Merchant. I think it was a very enlightening talk. We all learned a great deal. And you're absolutely right. All of this is a work in progress. We are all trying to do our part. And we learn more about the disease and more about our interventions with every passing day and with every passing week. Hopefully we become smarter by collecting data. I agree with Dr. Badullah that DRAP has been very active in and not only supporting the clinical trials, but also the pharmacovigilance process is also ramping up, and that's very good. We have played a very, very small part uh, in vaccinating people in Pakistan. Just for context, uh, the government has vaccinated more than 1 million people already in Pakistan. The private sector has vaccinated about 25,000 people. So while there has been a lot of noise about it, I want to be very clear that the public sector is really leading the way, and the private sector is going to play a supporting role. We are keen to play a supporting role, but it is a supporting role. Uh, as far as antibody levels are concerned, I completely agree with you what you said, Dr. Merchant, that uh, it is important to choose the right tests. Uh, right around the time when the CanSino trial was going on in Pakistan, there was a request from various physicians uh, to introduce anti-spike antibody tests in Pakistan. And we actually transitioned from antibody tests which were targeted against the nucleocapsid to antibody tests which were targeted against the spike protein. And so for the last 3 or 4 months, we've been using the anti-spike antibody test. We use two different tests. There is a Roche Diagnostics total antibody test, and there is a APPNA Diagnostics IgG quantitative antibody test. So, and both are available. Different physicians prefer different tests. But this is by all means uh, an ongoing, unfolding saga. And we are all in it together. We're all trying to do our part. And I think forums like these really inform us and help us deal with it in a more scientific, more evidence-driven manner. And I thank you again for this talk.
Thank you very much, Dr. Chaktai. Now over to Dr. Akhtar Hamid. We have some special your advice. What is the status of approval to use the COVID vaccine to the pediatric population? And what is going to be the, when can we see really something happening in this? Please, will you unmute your mic, please, Dr. Akhtar?
Thank you, Dr. Zair, and it's a very enlightening talk by Dr. Merchant. It's definitely a very unprecedented time, and the children are not being safe from there. If I could run 1 or 2 lines about what's going on especially in Canada, there is a total of 19% cases of all is positive for the children age 18 to 19. And in America is 13%. But last few weeks, this is going up to as up as 20% of all cases in children. And very recently, there is a data from Guelph University where they checked 220,000 cases, and out of those, 35% kids has no symptoms, and less than 9 years of age, and another 31% of children has only one single symptom, either a mild fever or just a one-off cough. So this is alarming as well, but this can spread it very fast as compared to, and another data is suggesting that adolescents are no lesser in spreading as other adults. So they are also equal to going with the virus spread. So the concern is this symptoms and this testing, which recently Ontario has opened up the testing in the schools. And what I have seen here, the 15,000 cases of children in school has been positive. while only 500 of the our teachers are positive. Having said that, still the Ontario government, especially in the Toronto region, they have opened up testing all the youngster 18 and above, either it's symptoms or not symptoms. Plus, they have also started the vaccination for them. And if I look at the status of vaccination, Pfizer has been already being already approved for 16 and above. In terms of 12 to 16 or 12 to 15 years, I think last 2 weeks ago, they were saying that they're gonna approve it, but I think it's still in the pipeline or they're just being approved. In terms of Moderna, they are doing a KidCove vaccination trial, and they are looking for 5 to 11 years, age of 5 to 11 years. And they have, this trial is also another with the vaccine is called 6 months to 12 years. So which is being Canada and in US. And in terms of AstraZeneca, I'm sure Dr. Merchant knows more, better because they were doing some trial on the age group between 6 to 17, but unfortunately there has been paused due to this hyped clot, reaction and whatnot. So so what we are seeing here that the children need to be vaccinated pretty soon, and the rate of vaccination is growing. The death rate is luckily is very low. So far, age 19 is only 8 deaths here in Canada, and in US is 2 deaths per 1,000 in 43 states they have reported. So from that point of view, these things needs to be speeded up, and I think the Canadian government, especially the Health Canada, is looking all the data very closely because there's a huge number of data to give you safety, especially since the AstraZeneca thing has come out. People are very cautious from that point of view. Thank you.
Thank you, Dr. Dr. Sarwar, do you have something to add, please? Please unmute your mic.
Yes, I have a very quick question, probably unrelated. What is the status of vaccine production in Pakistan? Whether there is any effort or we think it's far too above our capability?
Yeah, I would try to respond to your question. Historically, Pakistan had some facility for the basic vaccine manufacturing way back in 1980s. And surprisingly enough, we were among some of the few in Asian continent. But unfortunately, over the period of time, it died down. And there is just some very limited filling activity so far. But now, hopefully from next month, they are going to fill CanSino Bio vaccine, about 3 million doses. And the system is being prepared. But that is only a filling. So far, there is no more basic manufacturing facility. Like the Sputnik, they linked with 6 manufacturers in India to produce their vaccine, and the Sinopharm, they linked with a UAE group, and I think their first batch is going to come in the 4th quarter. But unfortunately, other than this filling, no other serious manufacturing vaccine facility. So that's all. Thank you very much to all of you. I think I really appreciate your patience. We passed the time. Generally, we close, but the topic is so interesting. The knowledge is so much. Still, we missed some of the question, and we will try to get the data of those questions and try to respond to you in the follow-up. So with this, we are thankful once again to Dr. Hamid Merchant for very knowledgeable presentation, Dr. Akhtar Ami, Dr. Chuktai, and all of the other panelists and the participants, the audience. Thank you very much. And have the remaining part of Sunday, but in the Pakistan, you have passed the Sunday, so it's night. So thank you very much once again, and see you in the next seminar. Thank you to all of you.
Thank you.
Thank you very much. Thank you.